Welcome. This is Lecture Notes Set 1 on lipids. The suggested readings from Leninger are listed here from the 5th, 6th, and 7th editions, and you can optionally do the practice problems, which are also given here from the 5th, 6th, and 7th edition. In this outline, we'll be talking about storage lipids, structural lipids, and other lipids. So, lipids are a very diverse chemical group. When we're going to look at these molecules of lipids, we're going to find that they have some similarities to each other, but there are a lot of differences. But the most common property, or the main common property that they have, is that they are all insoluble in water. So I always say that lipids are kind of like a hate group. They all hate water molecules. Lipids consist of hydrocarbon chains and fused rings, and they can consist of completely hydrophobic molecules, or they can be amphipathic. So just letting you know what this term means, amphipathic. Amphipathic means that the molecule has a nonpolar and polar end. So we'll just write that down here in the notes. So when we say that it's amphipathic, it means that there's a distinct nonpolar and a polar end to that molecule, okay? So I'll just write down here distinct nonpolar and polar end. So what we're going to find is that lipids, they all have the common property of being hydrophobic. Therefore, they're soluble in organic solvents, not in water, right? So organic solvents like benzene, chloroform, alcohols, and ethers. So the first thing we're going to learn about is the fatty acid, which is abbreviated as Fa. So the basic formula of a saturated fatty acid is this over here. So it's CH3 with a bunch of CH2s repeated over and over again. And these CH2 groups are called methylene groups. And it has a common functional group called the carboxylic acid functional group, which is COOH. Whenever we're talking about saturated fatty acids, it means that there's no double bonds present in the fatty acid structure. So the reason why they're called saturated is because they're saturated with hydrogens. Every carbon is saturated with the maximum number of hydrogens. So most fatty acids consist anywhere from 4 to 36 carbons, so C4 to C36. However, the most biologically relevant examples of fatty acids range anywhere from carbon 12, so they have 12 carbons, to 24 carbons. Fatty acids themselves, just as the name suggests, are weakly acidic, because their pKa value for their carboxyl group is anywhere from 4.5 to 5. Now, in terms of fatty acids that you'll find inside of cells, you won't find that many just kind of lying around because they are hydrophobic, and cells have a cytoplasm that is aqueous. So normally, if you try and find fatty acids inside of a cell, they have to be complexed to a carrier molecule like albumin in order to get soluble, to be solubilized. So, for example, when you have fatty acids in cells, they'll bind to a fatty acid binding protein, or when you find fatty acids in the blood, they have to be bound to albumin, which is allowing them to be soluble in the aqueous bloodstream. So those are saturated fatty acids. Saturated fatty acids have a common nomenclature, which I want you to know, which is C sub N colon 0. So this part of the formula here tells you the number of carbons that are in the structure, and this number over here represents the number of double bonds, which of course is going to be 0 for saturated fatty acids. And note that this nomenclature only applies to unbranched fatty acids, okay? So when we're talking about unbranched fatty acids, we're talking about something like this, okay? So there's no branches on this fatty acid over here, but if we're talking about branched fatty acids, so this is unbranched. And then if we're talking about branched fatty acids, it might look something like this. So those are methyl groups sticking off of some of the carbons over here, like I have highlighted in yellow here, and those would be branching points, okay? So in this course, we're really only concerned about unbranched fatty acids, and we're going to have one example of branched, but most of them are going to be unbranched. Okay, the nomenclature for unsaturated fatty acids. So if a fatty acid has a double bond into it, it's called unsaturated. and it follows this formula. It goes C sub n colon x, and this x represents the number of double bonds in the structure, so it has to be 1 or greater. Then you designate what type of double bond it is, so if it's cis or if it's trans, and then you get dash and then delta, and then y represents the position where the double bond is going to take place, and that's going to be the position relative to the carboxyl group, because we give the lowest carbon, like carbon number 1, priority, which is the carboxyl carbon. Okay, so if that doesn't make sense to you right now, we're going to go over some examples of this so it makes it very clear. Okay, so let's answer the questions here to learn about saturated, unsaturated fatty acids. So in question 1, it says draw the structural formula of fatty acid C18(0). Okay, so this is question 1. So we're trying to draw C18(0). So we need to draw the number of carbons here as 18. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Okay, and so we're going to put the carboxyl group over here. So this is carbon 1. This over here is carbon 18. So let's just double check. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Whoops. We are missing one there. There we go. That's carbon 18. Okay, so that's C18(0). This is a saturated fatty acid. And it wants us to draw the dominant species at pH 2. Okay, so at pH 2, okay, the first thing to note is that the pH is less than the pKa. So at pH 2, think of the pH as being a measurement of the hydrogen ion concentration. So when you have a low pH, it means that the hydrogen ion concentration is high. So therefore, the carboxyl group is going to be most likely protonated. Okay, so at pH 2, I'm just going to write down that the pH is less than the pKa. Okay, so in terms of the carboxyl group, it's going to favor COOH. When the pH is greater than the pKa, we're going to see COO- as what's called the dominant species. Okay, so this should be review from your Bio 201 or your Biochem 202 there, okay? So that means in A, we have to draw this as being the protonated form here. So I'm just going to change the atom. So this is protonated, okay? Because the pH is less than the pKa. So the dominant species is going to show a protonation on the carboxyl group. All right, next we're going to do B. So in this case, it says the pH is 7.4. So in this case, the pH is greater than the pKa. Because what was the pKa of the fatty acid again? It was 4.5 to 5. So here the pH is greater than the pKa for the carboxylic acid. So it's going to be COO-. So we draw this out. Let's just do our carbons again. So we're going to have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. And there we go. All right, so that is the dominant species at pH 7.4. Okay, question 2. So we're going to question 2 over here. And it's saying to draw the structural formula of the fatty acid C18(1) trans delta 9 at pH 7.4. So let's draw the structure out. So this is question 2. Remember, we're at pH equals to 7.4, so we're greater than the pKa. So this is going to be carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So this is carbon 1 again. 2, 3, 4, 5, 6, 7, 8, 9. So this is 9. This is 10. So the double bond is going to happen here. And this is a trans double bond. Right? Why is it a trans double bond? It's because the carbons are on opposite sides of the plane of the double bond, right? So it's a trans double bond. So this is going to be C18 1 trans delta 9. Okay? And we're at pH 7.4, so this carboxyl group is going to be deprotonated for the dominant species, okay? So that's a trans double bond. And then what happens with cis? So we're going to draw out, for question 3, the fatty acid C18-1 cis delta 9. So again, we're going to go 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yeah, so that's 10, 11, 12, 13, 14, 15, 16, 17, 18. Okay, so this is deprotonated there, so that's C1. This is 9, this is 10, and this is 18. So 1, 2, 3, 4. I always just double check these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. That's right. Okay, so this is a cis double bond. So just abbreviated DB is double bond because the R groups are coming off of the same side of the plane of the double bond. Okay, and this carbon 1 is going to be deprotonated once again because we're at pH 7.4. So this structure I just drew is called C18-1 cis and then delta 9. Okay, so hopefully that makes sense. That answers questions 1, 2, 3. Now, nomenclature-wise, right, we understand this thing now in the left column. The carbon skeleton tells you the number of carbons that are in the chain of the fatty acid, and then this number on the right tells you the number of double bonds, which is 0. Now, each fatty acid actually has a common name to it, and this is what you normally find like when you read ingredient packages, because the normal public, when they see fatty acids in food, this doesn't really mean anything to them. So the common name is going to be, like, for example, lauric acid corresponds to C12, myristic acid corresponds to C14, palmitic acid is C16-0, etc. Okay? Now, I don't want you to memorize these common names. They're just for your interest. So in case you do see these, for example, in some kind of food product, you know that they correspond to a saturated fatty acid. And there's also a separate set of nomenclature for unsaturated fatty acids. So for example, like C18-1 cis delta 9, its common name is called oleic acid. Or sometimes when you see C18:2 cis delta 912, that corresponds to linoleic acid, which comes from flaxseed. Okay, so again, anything with stars there, you don't need to memorize the common names, but you do have to know how to understand this notation, right? The number of carbons in the structure, the number of double bonds, whether it's cis or trans, and where that double bond lies in the chemical structure, okay? So make sure you know that nomenclature really well. All right. So that's hopefully a good introduction now to saturated and unsaturated fatty acids. We're going to now talk about the biological reasonings on why we're studying this. So most unsaturated fatty acids that you find from most biological sources are going to be in the cis form. So relatively speaking, trans double bonds are rare in nature. But we can get trans double bonds from different animals and an industrial process that's called hydrogenation. So ruminants, for example, like cows or sheep, in their gut, they possess bacteria that can produce trans unsaturated fatty acids through the process of fermentation. Okay, so in the gut of the cow, right, they have bacteria that can go on to make trans fatty acids. That means people who consume dairy products or meat from ruminants like a cow or a sheep, this is a source of trans unsaturated fatty acids in your diet. And also hydrogenation is a second source of unsaturated fatty acids. So margarine, for example, if you buy, for example, like Baicel is based off of plant oils, and plant oils have a lot of double bonds in them. So when you consume plant oils, which are largely unsaturated, hydrogenation, which is the process of adding hydrogen across the double bond, will cause the unsaturated fatty acid to become saturated, essentially. So that's what the process of margarine making is like, right? You take cheap plant oils, which are unsaturated, you hydrogenate them to make them into saturated fatty acids like they are in animal fats. Okay, so people who eat a lot of margarines, for example, that may not have complete hydrogenation, this is another source of trans double bonds in your diet. And when people consume trans fats, it's been correlated with an increased risk of atherosclerosis, which is the hardening of the arteries. And so that's why a lot of restaurants, for example, and grocery stores have made this movement towards less trans fats in their foods. Okay, so that's unsaturated fatty acids. Another term that you might have heard before is this term called the omega fatty acids, right? So when you're eating a yogurt, for example, or eating fish, a lot of nutritionists will say, oh, this is where you get your omega-3s, your omega-6s. And so we're going to talk about that notation. So when we talk about the omega notation, it just means that you're labeling it from the terminal carbon, right? The one at the very end. So what I'm showing you over here, this is C20:5 cis delta 5, 8, 11, 14, and 17. So just as the name suggests, there's 20 carbons in the structure. There's 5 double bonds that are all in the cis configuration, and they're found at locations 5, 8, 11, 14, and 17 there. Okay, but in the omega notation, the 20th carbon in this chain, which is the last one over here in our normal IUPAC notation, the last carbon, the 20th one, is the omega-1 carbon. Okay, so when we talk about omega fatty acids, we're going from the end instead, the terminal end instead. So if we go backwards, we can see that at the third position, going omega-1, omega-2, omega-3, this is where the first double bond is encountered. And so therefore, this fatty acid is called an omega-3 fatty acid. It has a common name called eicosapentaenoic acid, EPA, but it's an omega-3 fatty acid because the omega notation tells us that at the omega-3 carbon, this is where the cis double bond occurs. Now, the omega notation is not nearly as detailed as the CN:(X) notation, right? Because in this notation, it tells us exactly where the positions of the double bond is. But when we use omega, it just tells us where the first double bond is compared to the terminal end. It doesn't tell us the locations of other double bonds. So this is just called an omega-3 fatty acid, but we know the actual name is quite longer for the CN:(X) notation. So let's do another example. This is C20:4 cis delta 5, 8, 11, and 14. It's a polyunsaturated fatty acid, meaning that it has many double bonds. And what is the name of this omega fatty acid when we look at it? Well, this is the carboxylic acid carbon, and this is the terminal carbon over here, 20. So we count this as omega-1. We go omega-1, 2, 3, 4, 5, and the 6. This is omega-6. So we would call this structure over here, this would be an omega-6 fatty acid. Now, when nutritionists and dietitians tell you that you need to take your omega-3s, your omega-6s, they really are saying that you're taking these fatty acids, these polyunsaturated fatty acids, because they are what are called essential to the human diet. So that means we do not have enzymes that can make these fatty acids. And your body needs these fatty acids in order to do things like make molecules called eicosanoids, which you'll see at the end of this set of notes, okay? So if you do not get these fatty acids in your diet, you will not lead a healthy life. You may die as well. So that's why you need them in your diet. They're essential for you to survive. And so when we look at these fatty acids as well, they're typically labeled in common foods as being omega-6 or omega-3 fatty acids. And you do not need to memorize this either, okay? So I know this is like a really long chain, has a bunch of double bonds at different locations. Don't memorize this because that would just be painful, okay? Now, in terms of diet, in general, what happens is that people who consume an imbalance of omega-6s and omega-3s in their diet get correlated with an increased risk of cardiovascular disease. And so what I mean by that is the optimal dietary ratio of omega-6s to omega-3s in polyunsaturated fatty acids is about 1-to-1 or to 4-to-1. So you need more, in general, more omega-6s, or you want to keep this omega-6 ratio kind of low, right? You want to have this as a low number. So this is the optimal dietary ratio to have if you want to maintain a healthy diet. But in North America, especially in the US, the dietary ratio of omega-6s to omega-3s is about 10-to-1 or 30-to-1. Okay, so this becomes a lot bigger and this becomes a lot smaller. So that's usually because North Americans typically don't get enough omega-3s in their diets. You kind of want to increase this value to make the ratio more closer to this. Okay, so why is that? Well, where do we get omega-3s from in the first place? So you get them from leafy vegetables, from flaxseed oil, from different vegetable oils or hemp seeds, for instance, and you also get them from fish like salmon, mackerel, cod, and tuna. Now, many North Americans, they don't consume these foods. So this means that this ratio stays high because this number, the omega-3s, is quite low. So generally, you'll lead a healthier lifestyle if you consume a lot more vegetables, a lot more fish, in order to make this ratio closer to 1-to-1 to 4-to-1. Okay, if you have this ratio of polyunsaturated fatty acids in your diet, you are more prone to getting atherosclerosis, which can lead to plaques being formed in your blood vessels, leading to heart attacks or strokes. Okay. So let's talk about the physical properties of lipids as well. So, and especially fatty acids. You can see on the left here, this is the table comparing the carbon skeletons of fatty acids ranging from 12 carbons to 20. And you'll notice that when you have a homogeneous mixture, that means like only that specific fatty acid, it's assigned a certain melting point to it. So as you go from the top to the bottom over here, when you get to C20, your melting temperature increases quite dramatically. Okay, so the trend is that when you increase the number of carbons in the fatty acid chain, you get a larger melting temperature, melting point. So question 5 is, well, why is that the case? Why is there an increase in melting temperature as the number of carbons increase in the fatty acid chain? Okay, remember we're talking about homogeneous mixtures here. So if we had a whole mixture of C12 or a whole mixture of C20, you can see the melting point differs than more than 30 degrees. And so the reason behind this is because of what are called van der Waals forces. Okay, or London dispersion forces. You probably heard about those as well, right? So when you have hydrocarbon chains that are interacting with each other, they have instantaneous dipoles, which are going to attract. These are just some of the non-covalent interactions that happen in chemistry. And so when you have a longer carbon chain, you're going to increase the number of van der Waals forces, and that leads to more non-covalent interactions, causing the melting temperature to increase. Okay, so whenever you have an increased amount of van der Waals forces, you're going to get an increase in the melting temperature because it increases the overall stability. Okay, and that's when you increase the overall number of carbons in the fatty acid chain. Okay, so it's mostly van der Waals forces that we're talking about here. Okay, now let's look at one scenario here where we have a long chain fatty acid that has a differing amounts of unsaturation. So over here we have saturated, this is unsaturated. So trans delta 9 and a cis delta 9. So when we look at the melting temperature of C18, which is a saturated form, we get a melting point that's near 70 degrees Celsius. But as soon as you add in one double bond like the cis delta 9, you get this huge decrease in melting temperature. And so why is that the case? That's what question 6 is asking. And the reason behind this is because of what we call kinkiness. Okay, kinks. So unsaturated fatty acids are more kinky in that the carbon chain is going to kink outwards, okay? Don't get any other ideas in your head there. So let's go back over here to this diagram that I wrote to you in question 3. When we put in a cis double bond like this is considered to be kinky, right? It produced a kink in the chain and it's causing it to sort of move away in a different plane. And when you put a bunch of these unsaturated fatty acids together, they're not going to pack very closely together. They're not going to pack as well when you have these kinks in the chain. So that decreases the amount of van der Waals forces, and that's going to decrease the overall melting temperature. Okay, so we're going to say that when double bonds are introduced, this puts kinks in the chain. Right? It puts kinks in the chain, and therefore you're going to lower the amount of van der Waals forces. And this is due to less packing, right? So less packing together of fatty acids. And the overall result, when you lower those van der Waals forces, is it's going to decrease the melting temperature. Okay? So that's our answer to question 6 then. That's why we see a big decrease. And you can see that cis is more kinkier than trans. So this cis double bond makes a much bigger impact on the melting temperature compared to the trans, right? Because when we drew a trans double bond, it's kinky, right? But it's not as kinky. So it's just like less kinky when you have a trans double bond. But when you have a cis double bond, it's very kinky. And it's going to create a big kink in the chain, allowing less packing and less van der Waals forces. So I hope that makes sense. So going over here, right, these are saturated fatty acids. They pack nice and tight together like adjacent fatty acids. So that means the melting temperature is going to be high, right? Because there's lots of van der Waals forces in between all of these hydrocarbon chains. But as soon as you introduce those kinks, you get loose packing of the adjacent fatty acids, right? You can see these kinky chains. They do not touch as readily. They're not getting into that region where it qualifies as a van der Waals force, and therefore the melting temperature is going to go down when you have more unsaturated fatty acids. So my analogy to this is a lot like river dance, right? When you have river dance, you have all these dancers lined up next to each other. They're not social distancing right now, but if they this is pre-COVID, right? They're all dancing next to each other, and then all the saturated fatty acids are packed closely together, and this is going to create a lot of van der Waals forces in between each one of them, right? So they're going to have a high melting temperature. And so my reference here is to Beyonce's music video called Single Ladies, and a lot of you have probably heard of this song before, seen this music video. It's very famous back in the earlier 2000s. And so in this music video, Beyonce has 2 backup dancers, and when you kind of look at the structure of their legs as being hydrocarbons, it looks a lot like an unsaturated fatty acid chain. So let's just overlay this. There you go. So there's a bunch of fatty acids here. There's a bunch of unsaturation, and you can see that the legs are more kinked out. And there's more space in between them as well. So that means that when you have unsaturated fatty acids, you're going to get less packing, and you're going to lower the melting temperature. And as this video moves on, you can see this is like having a cis double bond, right? When you put a cis double bond in that chain, it kinks out even further, and then it's going to cause less packing and a lowering of the melting temperature. All right, so there you go. That is the Beyonce reference to there. So you can sing this song on your own. You can say, all the single bonds, and if you like it, then you better put a double bond on it. I think you get it. Okay, so let's do a practice question over here. This is question 7. So phytol. Okay, this is the structure of phytol. It's ester that you can find. Sorry, not ester. It's a long chain alcohol that you can find in plants. So this is phytol. And when you eat plant material, like let's say that you eat a lot of vegetables, you can get a lot of phytol in your digestive tract. Okay, so you eat the plants, you get lots of phytol into your body, and once it gets into your body, there's an enzyme that can convert this phytol into the structure that's called phytanic acid. Okay, so just to make this clear, you eat lots of this in vegetables, and then as soon as you eat a lot of this, your body converts it into phytanic acid. Okay, now phytanic acid is, when you look at it, it's a fatty acid, right? It's got a carboxylic acid group. It's got a long hydrocarbon chain, and it's a branched fatty acid, right? We went over that at the beginning of this set of notes. It's not unbranched, it's branched. Okay, and what happens is that if an individual has this disease, it's called Resum's disease. It's a neurological disorder, and it comes from the fact that this phytanic acid incorporates itself into the fatty acid, the phospholipid bilayer, okay, which phospholipids have fatty acids. We're going to see that in just a little bit, okay? And what happens is that when this phytanic acid gets incorporated into nerve cell membranes, okay, it's going to cause something bad to happen to them. So people with Resum's disease, they get this really big buildup of phytanic acid, and somehow when it accumulates in their membranes of the nerve cells, they start getting all of these strange symptoms that deal with their nervous system. So this was actually featured in House. It's a TV show, right? House. And there was a person in this episode where he consumed a lot of plant material, and he got this really big buildup of phytanic acid, and it caused destruction to the nerve cell membranes in his olfactory part of the brain, and therefore he wasn't able to smell properly. So what do you think would happen if you put this phytanic acid to a nerve cell membrane? Why do you think it's going to cause this effect? Okay, so this is kind of like a build-up concept of what we saw before with the unsaturated fatty acids. So if we had a bunch of phytanic acids all kind of packed together, do you think they're going to pack as nicely together as saturated fatty acids? And the answer to that is no, right? When you have saturated fatty acids, they're going to pack really closely together. But when you have a branched chain like this, they don't like packing close together. It's kind of like standing next to someone at a concert that has a bunch of spikes on their jacket, right? You don't want to get near them. So that's what happens with this phytanic acid, is that it's branched, okay? So we're going to write this down. This is question 7. So we're going to say that phytanic acid it's branched. Okay, so it's a branched fatty acid. And what happens is that when it gets into nerve cell membranes, it's going to cause it to almost liquefy, because it's going to decrease the overall melting temperature, okay? So we're going to write this down, that when phytanic acid is incorporated into membranes, it causes a decrease in the melting temperature, okay? And the reason is because there is less packing of fatty acids, which causes, which we know from unsaturated, a lowering amount of van der Waals forces. Okay, so that's why phytanic acid is bad. And what do nerve cells have, right? So nerve cells have this fatty layer, right? You might remember this from your physiology course, for example. But you have axon, right, which is going to have this insulation of fatty layer that's called a myelin sheath. And when you put these phytanic acid molecules into the myelin sheath, it's going to cause it to essentially liquefy, because the melting temperature of it's going to lower quite a bit. Okay, so we're just going to mention here that phytanic acid causes the destruction of the myelin sheath. Okay, so there you go. That's another concept that's kind of just an application of knowledge from the unsaturated fatty acids. Okay, our next concept over here we're going to talk about are micelles. So when you have free fatty acids and you put them into water, what happens is that they can form these structures that are called micelles, which are just basically these big spheres that are full of fatty acids, like millions of fatty acids into there, okay? And the reason why these micelles form is because when fatty acids are matched up, or when they get put into water, they form these conical-like structures. Okay, so water molecules, they really love that carboxyl group. Why? because the carboxyl group has charge and has a bunch of oxygen. So water loves it. So it kind of congregates all around here and makes this big circle. Whereas it hates this hydrocarbon chain. So when you put fatty acids into water, they're kind of like these little cone shapes that form as they interact with water molecules. And when you put all these cones together, they form these spherical structures that are called micelles, okay? So why do they form cones again? It's because the cross-sectional area when it's in water is going to be really big near the carboxyl group. And it's going to be really skinny near the hydrocarbon group. So it forms these little cones. You put all these cones together and they form these micelles. And micelles are quite interesting because this is what happens, for example, when you're using a detergent, right? Detergents are basically amphipathic molecules, meaning they have a nonpolar and a polar end, just like a fatty acid. And when you get all these detergent molecules together, they form these micelles. And what happens is when you're trying to clean, let's say, an oil stain off of your clothes, all the oil that's from the clothes is going to be bound to the hydrocarbon chain, like here, right? So all the oil stains are going to hang out with the hydrocarbon chains. And then the outside over here, all these polar head groups are going to interact and get washed away by the water. Okay, so with detergents, or in this case fatty acids, when you put them in water, they naturally form micelles, and micelles can solubilize oil droplets because the hydrocarbon chains love them, and then it's going to force the water to interact with the polar head groups instead. They're the polar fatty acids, carboxylic acid groups over there, okay? So that is the micelles, and again, they form, all these fatty acids form these little cone shapes. So here's another analogy to micelles, right? When you put a fatty acid, it's kind of like a pizza slice, right? And so you have the hydrophilic component over here, which is like the crust, and that's going to be where the carboxyl group is. And then this is the hydrocarbon chain, which is all hydrophobic and skinny, so it forms this kind of cone shape. When you put it all together, it forms a nice circle or sphere, which is going to be the micelle itself. Okay. So I hope that makes sense, that when fatty acids go into aqueous solution, they form micelles. Okay, the next thing we're looking at is the triacylglycerol, which is also known as the TAG, and this is a neutral fat. So TAGs are derived from what's called a glycerol backbone. So I want you to know this molecule really well, glycerol. Glycerol is a 3-carbon chain, and it has 3 hydroxyl groups, or it has a hydroxyl group on each carbon. And so what I call the glycerol is, I call it the parking space, the parking lot. Okay, this is like parking lot or parking space number 1, parking space number 2, and parking space number 3. And at each parking space, you can park a fatty acid onto it. Okay, so tags are made up of glycerol. This is glycerol over here. And a fatty acid can be added on to each of those positions on those hydroxyl groups there, okay? Now, when a fatty acid gets attached onto here, you'll notice that the entire fatty acid gets on and attaches onto the former hydroxyl group of glycerol. So what ends up resulting here is that you see that these are all ester linkages, okay? So this is an ester. And just to remind you, what is an ester again in organic chemistry? It's C double bond O to an O to one R group and then a second R group on that carbon over there. Okay, and we usually call that R prime, for example. Okay, so this is definitely an ester. So each of these fatty acids are linked to this glycerol and through ester linkages. And so tags, they're very reduced, meaning that they have a lot of electrons that can be potentially taken away from them, okay? Because you can think about all the different covalent bonds that are in the structure. And what you learned about in Biochem 202 and Biology 201 is that you can take electrons from a structure and then donate them to NADH or FADH2 to generate more energy through oxidative phosphorylation, okay? So when it says tags are very reduced, it just means that there's lots of electrons you can steal from here that could potentially be used in the electron transport chain to make more ATP. Now I want you to pay attention up here again to this is be able to draw and recognize the structure of a tag, right? I guess in online courses I can't ask you to draw it, but you should definitely be able to recognize the structure of tag because it's just glycerol, the parking spaces with 3 fatty acids attached to them there, okay? Now, what is the function of TAG? It's primarily used as a fuel source. Okay, so your body, when it has excess amounts of calories coming in from the food that you eat, your body is going to store more tags so that it's evolutionary like designed that in case you undergo starvation or famine, something like that, you're going to have lots of tags available because they can be used for energy if you are not consuming any food for a long period of time. Okay, so they're usually used as energy storage. And again, the carbons are really reduced, meaning that we're going to take a lot of electrons out of them. And they're more reduced compared to carbohydrates and proteins too, okay? So when something is reduced, it means it's rich with electrons and typically doesn't have a lot of bonds to oxygen, right? So when we look at TAG, you have tons of carbons bonded together, but not that many oxygens. Whereas you compare that to something like glucose. Let's just draw glucose for fun. Right, this is down. Whoops. Just make this look a little bit nicer over here. Okay, so this is glucose, and you can see there's a lot more bonds to oxygen over there, making it more oxidized. So we're going to get a lot more energy out of tags compared to carbohydrates or proteins. So in terms of fatty acids, right, if we take a look at their amount of energy you gain per gram, you get 9 kilocalories per gram if it's a fatty acid, whereas then you have carbs and proteins, which are not as reduced, right? They're more oxidized. You only get 4 kilocalories per gram. So you get more than double the amount of energy per gram of fatty acids than you would for carbohydrates and proteins. All right, so I hope that makes sense. And that's why your body is, in evolution, designed to carry a lot of fats in order to store a lot of energy for long-term reasons. Now, the other thing about tags is that when you store all that energy in the form of tags, you don't want it to carry a lot of water weight. So water hates triacylglycerol. That's why we call it a neutral fat. It's predominantly carbons and hydrogens, so water hates it. There's not a lot of polar covalent bonds here, so water hates it. Okay, so tags, they're very hydrophobic, and that means when you're carrying all of this tag around in your fat cells, there's not going to be a lot of water weight that's associated with them. And that's why, for example, if you're thinking about like your butt, for instance, which has tag in it, a lot of water is not associated with it, so our butts are not so droopy, if that helps you out, okay? So tags, they're very hydrophobic. They carry less water weight, and they tend to coalesce and be together because they're all hydrophobic into these dense droplets. So this is a white adipose tissue. This is fat tissue that's taken from humans, and each of these represents an adipose cell, or an adipocyte it's called. And these are all storing their tags in there, right? All these tags are just bunched up and grouped up in there, and they congregate in adipose tissue. And so a typical 70 kilogram person typically carries anywhere from around 100,000 kilocalories of tag, 25,000 kilocalories of protein, 600 kilocalories of glycogen, and 40 kilocalories of glucose. Now, don't memorize these numbers, but you can see the large majority of your kilocalories in a 70 kilogram person is occupied by tags. Okay. Now, tags, they take up about 16% of body weight on average. So that's pretty significant. Although, again, this varies depending on the person. If you're like a long distance runner, this number is going to be a lot smaller. 11 kilograms of tags can provide the energy needed for about a month. But the human's body only stores about a day's worth of energy in glycogen. So just so that makes sense, right? If you're undergoing starvation for a month and you have water, you can survive for a month because you have so many tags there, right? You have so much tag in your body that it's stored up that you can still produce adequate amounts of ATP. But you're only being supplied with about 24 hours worth of glycogen. Okay, so if you undergo starvation for a long time, yes, in the first 24 hours, you would get some energy from glycogen, but most of it's going to be coming from tags the other 29 days of the month. And like I said before, adipocytes, which are these fat cells, that's what that just means, is going to store the tags in there. So tags in adipose tissue can also be used for insulation at low temperatures, right? So for example, cute little animals like this one are going to have a lot of fat layers or adipose tissue that's going to contain all these tags because they're good for insulating body heat. So that's another function that tags can have. So hopefully I've given you a very good reasons why tags are important for your body. So let's answer some question 8. It's a conceptual question. It says, based on the structure of tags, why can this structure not form micelles even though fatty acids are a very major component of tags? Right? So this is the structure of tag again. And, well, why can we not form micelles with them? The reason is because once you connect these fatty acids to the glycerol, it loses its charge. That's a big one. Okay, so it attracts less water molecules. So we're going to write this down. So this is question 8, right? So tags, they're qualified, or they're called neutral fats. So they don't attract H2O. Okay, and therefore they are not amphipathic. And they can't form micelles. All right, and that fatty acid argument, right? When fatty acids attach to glycerol, it loses its negative charge in the ester bond. Okay, so water doesn't like it as much as soon as it's in that ester bond or ester linkage. Okay, and the last part of the question says, why are tags not major components of cell membranes either? Well, in order to be part of the cell membrane, you need to be a phospholipid, which is amphipathic, and tags are not amphipathic either, okay? So we'll just write down that tags are not amphipathic, but phospholipids are. Okay, and so hopefully that makes sense and emphasizes the point that tags are neutral fats. Okay, the last concept in storage lipids, we're going to talk about biological waxes. So make sure that you're able to recognize what the structure of a wax molecule looks like. A wax molecule is pretty simple. It's just a fatty acid. So this is palmitic acid, which is C16-0, and it's linked to a long chain alcohol. So this alcohol over here has 30 carbons, and this was formerly a alcohol group there, okay? So you have a long chain fatty acid in an ester linkage with a long chain alcohol, and that's what makes it a wax. And so as you can see, this is a really long hydrocarbon chain that's going to be saturated. So when you have large quantities of them and they're packing together, they're going to have really high melting temperatures. So that's why when you see like candles wax or beeswax, they have a really high melting temperature because they have really large number of van der Waals forces holding them together, okay? And wax can also be found on the surface of leaves, called a waxy cuticle, which helps to prevent water loss from the plant. So wax has a lot of purposes in everyday life. We get carnauba wax, which can be used to make candies like the outside of M&Ms, which makes them nice and shiny. And we also can use that carnauba wax for car wax as well. Thank you for your attention. This was the end of Set 1 Part A. Stay tuned for Set 1 Part B coming up next.